Hello again. Chapter 11, Lecture 3. We are cruising right along through genetics, and in this lecture we're going to talk in depth about dihybrid crosses and how the setting up of a monohybrid cross is going to be really the foundation on which we complete these problems. We also talked about Mendel and why he is now known as the father of genetics. We talked about pea plants and why pea plants make good test subjects. But for now, we're going to get right into kind of the foundation for all of genetics, and that is gametes. We've talked in depth about gametes and the production of gametes. Remember that meiosis is the process in which we produce gametes. Males are going to produce sperm cells, and females are going to produce eggs. And each of these gametes are only going to contain one copy of the chromosomes, which is why they're considered haploid. Right? They're only going to carry one allele per trait, and what that means is the diploid cell that begins meiosis will have two alleles per trait, just like all other diploid cells in your body. But after going through the process of meiosis and dividing that original diploid cell into 4 haploid cells, we end up with one allele per trait in both the sperm and egg cell, and that way when they bind at conception, both of these alleles make the set of 2 alleles per trait that zygote has, and then that fetus has, and then that baby has, and then ultimately that adult has. So sperm cell, egg cell both contain one allele per trait. They combine into one zygote, which has 2 alleles per trait. So let's get into a couple of our dihybrid examples. Now, hopefully you remember how we set up the monohybrid cross examples, because these are going to be very similar. We're just going to be looking at 2 traits instead of 1. And so the question is, in tomatoes, red fruit color is dominant to yellow, and we suppose that a tomato plant homozygous for red colors crossed with one homozygous for yellow. A, determine the genotype and phenotype of the F1 offspring. Okay, and so this is the question. This is kind of the setup of the question. This is the actual question. We want to determine the genotype and phenotype of the F1 generation. So these would be the parents. The F1 would be the offspring we produce from that cross. And so the first thing you want to do is we want to assign letters to represent our alleles. And so I have assigned the big R allele for red and the little r for yellow. Okay, and so I color-coded this to make it a little easier to understand or a little easier to follow. But it says in the problem, I haven't pulled out anything that the problem hasn't already told you. Red fruit color is dominant to yellow, so big R is red, little r is yellow. And now we just need to assign our parent genotypes. And so we have a red homozygous, or a homozygous red plant, and we're crossing it with a homozygous yellow. And so this would be our homozygous red parent, and we're crossing it with a homozygous yellow parent. Okay, maybe this is the male, maybe this is the female, it doesn't really matter. All we know is that there is one parent that is homozygous red and one parent that is homozygous yellow. The first thing we want to do is we want to segregate our alleles, and so in order to do that we draw a line and we determine that this parent is only going to produce a big R allele, because 1/2 of the time it's going to produce a big R, the other half of the time it's going to produce this big R, and so 100% of the time it's going to produce a big R. Over here, 100% of the time it's going to produce a little r. And so we end up with 100% of the time we produce a big R, little r, or a heterozygous individual from these 2 parents. Okay, and so your answer is, remember we want to find the genotype and phenotype of the F1 generation. 100% big R, little r is our genotypic answer, and 100% red is our phenotypic answer. Okay, so this is a little repeat or a little review of a monohybrid cross. Remember that is monohybrid because we're only looking at one trait. But I wanted to throw in a monohybrid cross example just as a warm-up so that when we move into dihybrid, it's a little easier to understand. Okay. So if we were to allow the F1 generation to self-pollinate, what would we expect the genotypic and phenotypic ratios to be? Again, this is an extension of that first problem. And so now we're taking these F1, because this would be our F1, this would be our parental or P generation. So we're taking two of these and now we're crossing them together. Okay? So we take a male of that generation and we cross it with a female of that generation. First thing you want to do, just like we did before, is we segregate out our alleles. And so half the time, or 50% of the time, we produce a big R. 50% of the time we produce a little r. Same thing over here. 50% of the time we produce a big R. 50% of the time we produce a little r. Now, what I like to do is I take these two options and I line them up on my page vertically. And so we take this and we throw it over here. And then we take this and we throw it over here. And these are the two options that will be produced from this individual. Maybe this is the male. Let's just assume this is male. And we'll assume that this is female. And so we take our two male options and line them up on our page. Then you're going to branch off of each of those two male options with our two female options. And so if I change my ink color and I take these two female options and I throw them over here, 1/2 big R, 1/2 little r. Again, throw these two options over here, and we get 1/2 big R, 1/2 little r. Then you combine them. 1/2 times 1/2 gives us 1/4 big R, gives us 1/4 big R, big R. 1/2 times 1/2 gives us 1/4 big R, little r. 1/2 times 1/2 gives us 1/4 big R, little r. Now notice the little r comes first in this case, but because our big R is dominant to the little r, we write it first. And to finish it up, 1/2 times 1/2 is 1/4, and little r, you can combine those. And our answer, again, because we're looking at genotypic and phenotypic ratios, is a 1:2:1 genotypic ratio and a 3:1 phenotypic ratio. What is that 1:2:1 and 3:1? 25% of our offspring, and these are the offspring, 1/4 or 25% are big R, 50% big R, little r, because we can combine those, and 25% would be little r. Okay, so that's how we get our 1:2:1 genotypic ratio. Phenotypically is 3:1 because, and I can change my ink color again, if I go back to red, these three all have at least one big R, and so it is going to be red. This one doesn't contain a big R, and so it's going to be yellow. So 3/4 of our offspring are going to be red, or 75%. 1/4 of our offspring are going to be yellow, or 25%. Okay? Now, if we took the red-fruited tomato plant, F1, and crossed it with the yellow parent, what might we expect the resulting offspring genotypic and phenotypic ratios to be? Again, we take our F1 offspring and we cross it with a yellow parent. This, you know, has to be yellow because it doesn't contain a big R. This is a big R, and we know that this is the genotype that is produced by the F1, or by the parent, so it is in the F1. Segregate out our alleles, so again we're going to have 1/2 big R, 1/2 little r. Over here, 100% are going to be little r, so this little r is going to be passed on half the time. This little r is going to be passed on half the time. Again, we're going to throw our male options over here, and so 1/2 big R, 1/2 little r. We're going to then combine our two female options. Again, we can consolidate if we want, otherwise keep them separate. And so 1/2 is going to get this little r, 1/2 is going to get this little r, 1/2 little r, 1/2 little r. Now you combine them, and so 1/2 times 1/2 is 1/4 big R, little r. 1/2 times 1/2 is 1/4 big R, little r. 1/2 times 1/2, 1/4 little. 1/2 times 1/2, 1/4 little. And so if you can combine those, notice that these are the same. These are the same. And so our genotypic ratio would be a 1:1, 50% big R, little r, 50% big R. And our phenotypic ratio is 1:1 as well, where 50% of our offspring are going to be red, 50% of our offspring are going to be yellow. Okay, again, notice the difference between the genotypic and phenotypic ratios. Genotypic is the genetic component, phenotypic is what they physically look like. Okay, so those are some warm-up monohybrid cross examples. Hopefully those are review for you. And so let's get into what the independent assortment or principle of independent assortment is. The independent assortment principle is a Mendelian principle discovered prior to the knowledge of meiosis, yet it describes the process of meiosis to a T because it talks about the separation of chromosomes being completely random, which we know happens during synapsis when those homologous chromosomes come together. We know that how they come together is completely random, and the first pair of homologous chromosomes doesn't have any bearing on the second pair, and those first two pairs don't have any bearing on the third pair and fourth pair, and so on. So if we get into what this dihybrid example looks like, you'll notice that now we have two alleles. So we've got, or two sets of alleles. We've got a gene and a B gene. We have two alleles for each gene. So we've got a big A allele, small a allele for the A gene. We've got a big B allele and a little b allele for the B gene. And now we want to see how this independent assortment or principle of independent assortment will segregate out these alleles, because if you remember from the monohybrid example, all we had to do was draw those arrows straight down and separate our big A from our little a. And we said half the time the big A will be passed on, half the time the little a will be passed on. Now we are combining the A and B genes as a unit, and so we can't just separate those out like that. We have to FOIL. Okay, and so a lot of times in math class, you'll see or use the distribution principle or FOIL, sometimes it's called. And so you'll notice that in this case, the first gamete produced is a combination of the first allele of each group. So if this is a group and this is a group, we go first, which is going to give us 1/4 or 1/4 big A, big B. Then you go outside, right? FOIL, first, outer, inner, last. So first is big A, big B. Outside is big A, little b. Inside is little a, big B. And last is little a, little b. And so you'll notice that each of these options is equally likely to be passed on. This could be a sperm cell, 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 or they could all be eggs. But the unique thing is that it was discovered that each of these allele groups will be passed on in equal concentrations. Even if receiving both dominant alleles makes you a more fit organism, doesn't mean that these won't happen at the same or equal frequency. They all happen at the same frequency. The second way you could go about doing this, if you're not a fan of the FOIL method, is you can basically separate out the A's and then separate out the B's and then do a little lattice of sorts. And so if the big A, if we know from our monohybrid cross examples that the big A is going to be passed on half the time and the little a is going to be passed on half the time, put them down just kind of in a vertical way. Then the same is true for the B's, where big B is passed on half the time and little b is passed on half the time. Then just kind of combine them all. Combine this one with this one and we get 1/4, because 1/2 times 1/2 is 1/4, big A, big B. Then you combine this one with this one, 1/2 times 1/2 is 1/4, big A, little b. Then combine this one, half times half is 1/4, little a, big B. Again, we're going to put little a first because big B is not dominant to the little a, and A's have always gone first, and so you keep that trend the same. And then our last one is half times a half is 1/4, and it's little a, little b. Notice that the distribution of gametes in this case is identical to the gametes in this case. So it doesn't matter which method you use to segregate out the alleles for a dihybrid cross, just know that it is required that you know how the gametes are produced and how the alleles segregate themselves out. And either of these will do. So, an example. If we cross a tall purple plant, and we're not talking about people because people are not purple, but if we cross a tall purple pea plant with a short white pea plant, if you remember from the the pea plant characteristics or the traits that were easily identifiable. Tall versus short was one of them that Mendel looked at, and purple versus white was one of them that he looked at also. And so we're going to assume that tall and purple are both dominant. I'm using big T to represent tall, little t to represent short, big P to represent purple, little p to represent white. And if we cross these two individuals, whether this is the male or this is the male, doesn't matter. One is male, one is female. We first have to separate out our alleles and come up with our gametes. And so if we FOIL, just like we did, we go first, outer, inner, last. Basically, these are the gametes we come up with. Okay, so if we go first, outer, inner, last, every single one of the options is going to be identical, right? 1/4 of the time we're going to get a big T, big P. 1/4 of the time we're going to get a big T, big P. The other 1/4 of the time we're going to get big T, big P, and so on. So every single one of the gametes is going to be big T, big P in this example. So we say 4 out of 4 are big T, big P. In this case, same thing happens, right? We FOIL this out and we notice that every single one of their gametes, in fact 4 out of 4 or 100%, are going to be little t, little p. When you cross a 4/4 times a 4/4, you get a 16/16 or 100% are heterozygous, meaning big T, big P, little p, and you get 100% heterozygous. That is your genotype. What is the phenotype? 100% tall, purple. Okay, 100% tall, purple. So if we cross this plant with this plant, we get 100% tall, purple plants. But these tall purple plants differ from the tall purple parent because these tall purple plants carry a recessive allele for each of the traits, whereas this parent does not carry the recessive alleles. These recessive alleles came from this recessive parent, but they are not expressed because they're covered up or dominated by the dominant alleles given by this parent. A little more extensive example would be when we take 2 of those tall purple plants we just produced in the F1, and now we cross a tall purple heterozygous plant with a tall purple heterozygous plant. Okay, notice that we are heterozygous for both the T trait or the T gene and the P gene. We're going to find our gametes by FOILing. So first would be big T, big P. Outside would be big T, little p. Inside would be little t, big P. And last would be little t, big P. And you'll notice that these gametes over here would be identical to the first parent because these are identical people or identical pea plants because they were produced in the same offspring or generation. Okay, these would have been produced in the F1. We are looking for results for our second filial or F2. The easiest way to do this, and I'm not showing you how to do a Punnett square, I'm just kind of showing you how to do a branching, right? If these are the gametes that are produced by this individual, okay, let's assume maybe that this is our male individual and this is our female individual. I don't know why I just did that, but this is our female, this is our male. These would be the sperm cells, these would be the egg cells, and so if we start combining these, 1/4 times 1/4 gives us 1/16 big T, big T, big P, big P, and so you see that right here, 1/16 big T, big T, big P, big P. Then we combine this one with this one. We get 1/16 big T, big T, big P, little p. Then we combine this one with this one, and we get 1/16 big T, little t, big P, big P. And then we combine this one with this one, and that finishes out our first male gamete combinations with a big T, little t, big P, big P. Now I'm going to switch the colors, and we're going to keep going. And so if we cross this gamete with this one, because again we're trying to get all possible combinations, you will get 1/16 big T, big T, big P, little p. Cross with this one, we get a big T, little p, little p. Cross this one with this one, and we get a big T, big P, little p, or a heterozygous individual. We cross this one with our last one, and we get a big T, little t, big, back up. big T, little p, little p. I shouldn't have picked letters that rhymed, T's and P's. If we cross this one with our first female option, we're going to get big T, big P, big P. Cross this with our second female option, and we get big T, big P, little p. Cross this one with our third option, we get little t, big P, big P. And keep going, we get a little t, big P, little p. Now we cross our last male gamete with our first female gamete, and we get a 1/16 big T, big P, little p, which is heterozygous. Cross last male with the second female gamete, and we get big T, big little p, little p. Keep going, and you get little t, big P, little p. And our last option would be little t, little p, little p. Now notice every single one of these is 1/16 because there are 4 male options, and we're timesing those or crossing those with 4 female options, and so there are 16 possible combinations. There's 16 different lines here, and when you combine those all, you get 1/16 for every single one of those. But hopefully you noticed as we went through there that some of these are repeats. In fact, here's a heterozygous individual, here's a heterozygous individual, here's a heterozygous individual, and here's a heterozygous individual. So we want to consolidate these and make it a little easier to understand. And so what we can do is we can start consolidating. In fact, we've got 1/16 homozygous dominant for every single one, right? So now we can cross this one off because it's here. Then we've got 2/16 big T, big P, little p. And so you'll see 1/16 here, and there's another one right here, so we can cross that one off. Those are the same, so we'll put those together. So it's 2/16. We don't need 2 of the same ones written twice. We'll just put 2/16 that. Then we've got 1/16 big T, little p, and so you'll see that here, and we can cross that one off, right? You'll notice that this is a 1 to 2 to 1 ratio, but we can keep going, keep consolidating. Big T, big P, big P. So you'll see 1 is here, and you'll see 1 here. 4/16 are those heterozygous, the ones that I pointed out to begin with, right? So here's a heterozygous, here's a heterozygous, here's a heterozygous, and here's a heterozygous. 4 of them. 2/16 are going to be heterozygous for T, but homozygous recessive for P. So you'll see 1 there, and 1 here. 1/16 is going to be homozygous recessive for T's and homozygous dominant for P's. So there's 1 right here. 2/16 are going to be homozygous recessive for T and heterozygous for P. So there's 1, 2. And our very last one is 1/16 is going to be little t, big P, or little p, or homozygous recessive for all, or both traits. Now, we've just taken all of these options and we consolidated them to form our genotypic ratio. And the genotypic ratio always will be for 2 heterozygous individual, dihybrid individuals, is going to be 1 to 2 to 1, 2, 4, 2, 1, 2, 1. Okay, 1, 2, 1, 2, 4, 2, 1, 2, 1. If we consolidate these and come up with our phenotypic ratio, because again, this is genotypic ratio, phenotypic ratio is going to be 9/16 are going to be tall purple, because again, tall and purple are our 2 dominant characteristics, dominant traits, and so these are going to be tall purple, tall purple, tall purple, tall purple, and so that would come up with 4 plus 2 makes 6, 7, 8, 9. 9/16 are going to be tall purple. 3/16 are going to be tall white. Tall is when they have at least 1 dominant T, so this one's going to be white, tall and white. This one's going to be tall and white. And so 2/16 plus 1/16 is going to give us 3/16 or tall and white. 3/16 are going to be short and purple. This one's short and purple. This one's short and purple. 2 plus 1 gives us 3/16 short and purple. 1/16 is the only one left, and this one is short and white. And so the phenotypic ratio is 9/16, 3/16, 3/16, 1/16, or a 9/3/3/1. Genotypic ratio is a 1/2/1, 2/4/2, 1/2/1. 2 unrelated traits were crossed. If we crossed a parent that is round and yellow, or has that, and we cross that with an organism that is wrinkled, meaning they're recessive for all of them, or little r's and little y's. We cross this parent with this parent, we get 100% heterozygous organisms. Okay, that is a true dihybrid cross. And so when we take these heterozygous individuals and cross them with themselves, we get
9 yellow round, 3 green round, 3 yellow wrinkled, 1 green wrinkled.
Sorry about that. And so if we look at a Punnett square, hello, Punnett square. If we look at a Punnett square, which corresponds with these results, you will see what the P's look like at each particular box. And remember in class when I talked about a dihybrid Punnett square having 16 different boxes, this is an example of a dihybrid Punnett square with those 16 boxes. So remember when we come up with the alleles or the gametes, we had to FOIL these, and these are the combinations that we came up with. Okay, so we would have FOILed both of these. We would have come up with both of these gamete combinations, and then you just combine them. So big R, big Y gives you this combination. And I'm not going to go through all these boxes, but if you get to this one, little r, big Y, and this one, little r, big Y, you'll see that when you combine those, little r, big Y, you get a wrinkled yellowed P. This one would be the only one that is wrinkled green. You should notice that 1, 2, 3, 4, 5, 6, 7, 8, 9 of them are round and yellow. 3 of them are round and green. 3 of them are wrinkled yellow, and 1 is wrinkled and green. That's that 9-3-3-1 ratio we just came up with in the last slide. Okay, so think about this for a minute. I know that last problem was pretty long, pretty lengthy, when we had to come up with all the genotypes and then consolidate them and come up with the 1-2-1, 2-4-2, 1-2-1 genotypic ratio and the 9-3-3-1 phenotypic ratio. This is an easier way to do it. If the question tells you to come up with specifically the probability of round yellow offspring and they are asking you to come up with a phenotype, you can try to answer the question based on your understanding of the ratio of the phenotypes and not worry about coming up with any of the genotypes. For instance, if we want to come up with the probability of a round yellow offspring, and we know that when we do a monohybrid cross, 3/4 of our offspring are going to be round and 1/4 is going to be wrinkled, and we know that the probability of doing this monohybrid cross gives us 3/4 yellow and 1/4 green, because round and yellow were the two dominant traits, wrinkled and green being the two recessive traits. We can take 3/4 round, which is what we would get if we were just doing a monohybrid cross between round and wrinkled. Okay, 3/4 round, 1/4 wrinkled, and we can times these by our next phenotypic results, which would be 3/4 yellow and 1/4 green. If we use our arrows like we did in the beginning, or one of the beginning questions, we take 3/4 times 3/4. 3/4 times 3/4 gives us 9/16. 3 times 3 is 9. 4 times 4 is 16. 9/16 of them will be round and yellow. 3/4 times 1/4 gives us 3/16 will be round and green. 1/4 times 3/4 gives us 3/16 are going to be wrinkled and yellow. And 1/4 times 1/4 gives us 1/16 is going to be wrinkled and green. Okay. So all we're doing is we're taking our known phenotypic ratios, or our phenotypic outcomes, and we're timesing the known phenotypic outcomes of individual completely independent events. And this principle of independent assortment says, and the principle of probability, or the law of probability, states that two unrelated independent events will happen or can be combined basically as the sum total of their individual parts. And so 9-3-3-1 is that same phenotypic outcome, that same phenotypic result, or phenotypic ratio we can get by doing it this way. We don't have to do a Punnett square, and we don't have to do the big long drawn out process. Okay, so what is independent assortment? And I'm going to try to finish up in the next 20 seconds. Independent assortment, remember, is how the chromosomes line up during synapsis. Remember, synapsis is when the homologous chromosomes come together. This is a tetrad. This is a tetrad. This is just as likely of an orientation as this. The only difference is how they line up, and then ultimately how they separate out. Notice that this combination is different from this combination, which is different from this combination, which is different from this combination. And so independent assortment, along with segregation, is one of those vital processes that allows us to produce several different combinations of gametes, which is one of the biggest contributors to genetic diversity, regardless of what species you're talking about. Humans have a lot of genetic diversity. Animals have a lot of genetic diversity. One of the reasons, like I said before, is independent assortment and how these chromosomes line up during synapsis. And there would be more combinations than just these two, right? Because human diploid cells have 46 total chromosomes. And so you can imagine that there would be several different combinations that are a result of this independent assortment process when you have a total of 46 chromosomes. So that's all we're going to do for now. The last lecture of Chapter 11 is going to be all about non-Mendelian things like incomplete dominance, codominance, multiple alleles, sex-linked inheritance, and polygenic inheritance. And we're going to talk in depth about each one of those. So until then, see ya.